action potential, we have voltage-gated sodium. But in the heart, we have voltage-gated calcium. And these are fast-acting compared to others we'll see later. So these are called fast voltage-gated calcium channels. Calcium diffuses into the cell, positive charges, and so we see this sharp rise in the positive past zero, creating an action potential. And that current is going to flow through the autorhythmic cell, out of the autorhythmic cell via gap junctions into contractile cells, and from one contractile cell to the next. It does that same kind of domino effect. All right, as these fast calcium channels close at the top, similar to sodium, as we reach our peak of our action potential, then we have potassium voltage-gated channels. And those are going to allow potassium to leave the cell. Because it's going from its high concentration to its low concentration. As potassium ions leave the cell, the inside of the cell becomes more negative and repolarizes. Obviously, we need active transport pumps to move the sodium back out again, or it would just fill with sodium and swell it with water. Okay, so we have those are understood. We repolarize, and then instead of reaching a resting membrane, the cell automatically starts to depolarize again. Every time it reaches threshold, a heartbeat is triggered. Okay? Yeah?
How fast would you say it takes for the pulse to circulate throughout the myocardium itself?
Well, I'm not quite sure what you're asking.
From like the open creative potential.
It's fractions of a millisecond. So you'll see that with the ECGs and you find the components.
You know how in the movies they show, that shot that they give patients epinephrine? And does that trigger the cells to contract?
We'll get to that in just a few minutes.
Oh, okay.
Okay? Okay, safe. Now, based on what he asked, how do we change heart rate? This without nerve innervation, the SA node, and that's the group of autorhythmic cells found at the base of the superior vena cava. So we're talking about right up here. So these autorhythmic cells will depolarize on their own about 100 times a minute. Okay? So they're driving the heart rate. These are about 60. These here are about 40, and individual muscle cells in the atria and the ventricle, if left on their own, would be about 15 times. So obviously, if this one is 100, before they have a chance to depolarize on their own, they're going to be made to depolarize by the current coming from the SA node, kind of making them depolarize. Anybody here have a resting heart rate of 100? Fastest my heart rate has gotten is 210. That was climbing in the wilderness area up at about 7,000 feet. My husband thinks I'm totally out of shape because she's getting on the elliptical. I don't get to I'll regularly get to 170, 180. Do that for about 20 minutes. My husband's 59, and his heart rate's about 48. My 20-year-old son's heart rate is about the same, 46. So when he was in his senior year of high school a couple years ago, he needed an extra PE. Most of his PE was playing football and basketball and stuff, but he needed an extra semester. And so he took PE with the freshmen. And the coach was all about fitness, so he had these monitors. And to get an A in the class, you had to get your heart rate up to a certain level and keep it there for so long and show improvement over the semester. Well, he had a set number he wanted them to get their heart rate to, rather than a percentile, or so if they're instead of double your resting heart rate, they had to get to like 120 or 160. That's a little easier when you're starting at 80 than when you're starting at 45. So to get his heart rate up to 160, he had to work really, really, really hard. And I kept telling him, I said, why don't you, talk to your coach and say, look, doubling my heart rate would be 90. Doubling this freshman girl's heart rate would just be, 120 or something, or 160. So I'm having to work really, really, really hard. And he doesn't go and talk to his coaches, so he just not there with it. But we obviously have to do changes. So there is something that is slowing our heart rate down. And obviously we can have heart rates higher than 100. So we have the ability to speed our heart rate up. That's where the autonomic nerves come in. Okay? That's what we need our nervous system for. So someone who has had a heart transplant does not have the ability to have their heart respond to stress from the nervous system and within a few microseconds increase their heart rate. They have to get their norepinephrine from where? Adrenal glands. Okay? Do we have any endocrine organ that releases acetylcholine to slow the heart rate down? No. So they have to wait for the half-life of the norepinephrine in the bloodstream to be decreased by excretion or uptake or whatever, and that's much longer than it would be if it was just the nervous system. So let's move ahead and to some of the slides here and look at parasympathetic. This looks scarier. I'll take it through. It looks scarier than it really is. And there's no new information today, okay, other than what we've just, this stuff over here. This stuff you already know. As scary as that looks, you already know this. G protein, ever heard of that before? Alpha subunits, ever heard of that before? Okay, you don't have this in your lecture notes. I added this last semester. But the concept is there, so just bear with me. So, what would be the nerve that delivers acetylcholine to the heart? Within the heart muscle at the SA node. So this would be the SA node here. This would be the preganglionic fiber. And it's going to be delivering acetylcholine here in the ganglion and acetylcholine here at the SA receptors. So in the ganglion, well, first of all, what nerve is that? Heart. What nerve carries parasympathetic to the heart? Vagus. Back on familiar territory, okay? No more pelvic and sacrosplanchnics for a while. So vagus nerve. Acetylcholine here. Let me draw this a little bit bigger. Receptors on our postganglionic neuron are nicotinic. Remember hearing that word? Bio 430? Okay, same as the skeletal muscles. These receptors are sodium ion channels. So when acetylcholine binds to them, they open. And allow sodium ions into the cell. They're always excitatory. Sometimes they're called ionotropic because they're ion channels. They directly open. All right, this is our postganglionic neuron. It's inside the cardiac muscle tissue. And it's also releasing acetylcholine. But this is binding to muscarinic receptors. And these are always G proteins. Okay, so this would be an autorhythmic cell. So the vagus nerve fires, releases acetylcholine, which excites this neuron, always. It fires, releases acetylcholine, which inside the heart will bind acetylcholine to its receptor, which activates a G protein. So guys, okay, maybe you're not comfortable, but you're familiar with this. Two things happen, right? The alpha subunit actually turns off adenylate cyclase, and that causes voltage-gated channels to close. So it's going to cause some of these voltage-gated channels here to close. All right? So we're going to close them earlier and cut off some of the positive ions that are coming into the cell. The beta and the gamma subunits, which we haven't talked about doing much, are going to bind to voltage-gated potassium channels. They're going to open. And what does that mean? So the calcium channels are closing, not as much positive ions are coming into the cell. The potassium channels are opening. What are positive ions doing? They're going out. So we're trying to make the inside of the cell more positive, and yet we're losing positive ions. So let's say we're trying to fill a bathtub, and we've got a quart-sized container that we're adding water to the bathtub, okay? That would be our leaky sodium channels. And we fill it up and it overflows and we have an action potential. Well, now we're filling it up with our quart-sized container, but then we pull the plug. It's going to take a lot longer before it overflows, right? Because we're losing half of what we're adding. So that's what's happening here. With the potassium channels open, it's taking longer to gain enough sodium, enough positive ions to reach threshold. So what we see is this, the influence of acetylcholine. Every peak is a heartbeat. Our heart rate has decreased. So this is the primary reason why your resting heart rate is not 100 and is closer to 68 to 70. Now an athletic heart, okay, which is what my husband and my son have, means a stronger muscle. It doesn't make it contract and pump more blood with each contraction. It doesn't have to pump as often. So those types of events, a really weak heart has to pump faster. All right, so those type of events will have an effect as well. But just on the control here, acetylcholine is going to cause more potassium to leave, therefore it's going to take longer to reach threshold. And we'll have fewer action potentials for every autorhythmic cell. Okay. This, I'm not going to have you trace this or read it, but what it's saying is parasympathetic nerve is sending vagus, which is releasing acetylcholine and binding to a muscarinic receptor, which is an inhibitory G protein. That's what the I there means. Okay? It's going to block adenylate cyclase and open calcium, going to cause the calcium channels to close, and I don't have the potassium channels on there, but that's all that's saying. As you can see here, it's causing it to not reach, because the calcium channels have closed, it's not going to become as high number of positive ions inside when it does reach action potential, and it's going to take longer to actually reach the action potential. Okay? That would be the effect of acetylcholine. And we have it happening in homeostasis. That's what keeps our resting heart rate lower. Okay? Any questions on that? It's new concepts, but using older information, just putting it together to explain newer events. All right, so now let's go back to this one. So let's say now that I'm starting to get stressed, or I'm starting to exercise and I'm sending sensory fibers up to my brain, and it's going to start activating the sympathetic nervous system. So just a little bit of anatomy. We're not talking about splanchnic nerves here. You guys remember the sympathetic chain? All right. When we get to the neck region, they kind of combine and form just three. One at the clavicle, one here about your jawline, and one up right close to the neck. So we just have three spherical ganglia. The lower one and the middle one will send fibers to the heart. They're called cardiac accelerator nerves, and that's it. Okay? So you can tell the difference between vagus and cardiac accelerator nerve, I think. So let's change this. Can I change this here? Let me just kind of add to it what's happening. So this is going to cause calcium channels to close, potassium channels to open. Okay, this is what I had up on the board, but kind of closing off the summary of what's occurring there. The activation of the G protein. Now we need to speed up our heart rate. Notice it's going slightly higher than what we have with just our regular autorhythmic cells, and it's occurring more often. So we're getting positive ions, more positive ions into the cell, and a stronger current when we do reach our action potential. That's not quite as important for the autorhythmic cells. Can I erase this now? Okay. Actually, all I need to erase is the word and this part. So this is now going to be sympathetic color. And these will be cardiac, accelerator nerves. This, by the way, is still acetylcholine. This is still a nicotinic receptor, even though this is sympathetic. Okay? And then, this is going to come to our, I'll make it shorter so those of you in the back can see it. This is going to come to two different types of cells. In the SA node, it's going to come to cardiac autorhythmic cells. And it's also going to come to contractile cells. We'll look at those later. Guess what? It's a G protein again. And these are identified as a beta 1 receptor. And you saw this already. Let me just take a moment and pull up this picture that you have of this. It's in the endocrine lecture, and it was an example. neurotransmitter. Epinephrine tends to be the neurohormone. Binding to its beta-1 receptor, which is associated with the G protein. When activated, activates cyclic AMP, and that's going to open up calcium channels, both on the cell membrane and inside the cell on sarcoplasmic reticulum. This part is more important for the contractile cells, okay? This part is important for our autorhythmic cells. But this is a pathway that you're already familiar with, the word cyclic AMP. So norepinephrine is binding to beta-1 receptors. The alpha subunit activates adenylate cyclase. Adenylate cyclase activates ATP to cyclic AMP. Cyclic AMP binds to voltage-gated calcium channels. Nothing new. We're just applying it here. All right. So here is what I wanted. So here is norepinephrine binding to the beta-1. This time it's a stimulatory G protein rather than an inhibitory. So we're activating cyclic AMP instead of breaking it down. And the cyclic AMP is allowing calcium into the cell. Okay? So that additional calcium is going to increase the positive charges inside the cell. And we're going to reach our threshold potential earlier. So if we diagram that on the screen, rather on the whiteboard, it's going to take less time because we're adding positive ions in addition to the ones that are passing through the leaky sodium channels. And now we're increasing the number of times the cell creates an action potential and the number of times the heart beats in response to that. So anytime the heart rate goes up in response to this, it's because we're reaching our threshold earlier. Therefore, we're making more of them, more action potentials during any given period of time.
And that's basically because of the increasing calcium.
Right. So additional positive ions, being calcium ions, are added to our sodium. So we're back to the bathtub illustration. We're trying to fill our bathtub with the quart container, which would be our leaky sodium channels. And now instead of pulling the plug, we pick up another quart container, and we use both of them. So we're going to reach overflowing earlier. Okay? So we have a faster heart rate. Now we'll see those additional calcium ions when we get to our contractile muscle cells will have a different effect, all right, as far as the strength of the heart's contraction. But that's the effect on heart rate. So this diagram now, we're moving to cardiac contractile cells. So we've reviewed skeletal. We've done cardiac, autorhythmic. I'm going to leave this up here for now. Because on the quizzes and on the exam, remember how I told you there'd be certain things on your lecture exam? And they were there, right? So certain things will be on your heart test and they will be there. You just can't get around them. It's only there's questions that are so good you just have to use them every time. So you will be asked to identify what a particular membrane potential pattern shows. So if we have a membrane potential pattern, that's skeletal muscle, okay? With the resting membrane potential, a graded potential, an action potential, and back to a resting membrane potential. That's cardiac. Autorhythmic. No resting membrane potential, but a graded potential and action potentials. Okay, and then what we're going to look at now is the cardiac contractile cell. A resting membrane potential, an action potential, no graded potential. For each of those, you should be able to identify what channel is creating those events. So, for skeletal muscle and the resting membrane potential, we talked about the 3 conditions. Leaky potassium channels are more than leaky sodium channels. The active transport pump pumping out more sodium than it's pumping in potassium. And the negatively charged proteins create our resting membrane potential. And that's same for cardiac contractile cells. Okay? What causes the graded potential in skeletal muscle? What kind of channel opens? Remember the list of channels I wrote over here on the board? Leaky is going to be involved here. What's going to cause that? Ligangated. When acetylcholine binds to its receptor. Okay? For our action potential, these are caused by voltage-gated, sodium, and potassium. All right? Okay? Cardiac autorhythmic cells. What causes our graded potential? Leaky sodium channels. We can change it by the number of voltage, potassium, and calcium, but what it's originally due to is leaky sodium, fast voltage-gated calcium, and voltage-gated potassium. All right? And now we're going to look at this. So be able to recognize the 3 different types, identify whether it's a graded potential or an action potential or a resting membrane potential, and then what channels are forming those potentials. Okay? Again, it shouldn't be something drastically different than what we covered in Bio 430. We're just looking at some new ways of application. All right. Can I erase this now? Because I want to save this part of it over here. You guys have these 3? We're going to look at this one now. You can go back and pull them out again. So, now we need our cardiac contractile cell to act like a skeletal muscle cell and shorten. So when the atrial muscle or the ventricular muscle is not contracting, it's relaxed. That's why we have a resting membrane potential. Autorhythmic cells never have a resting membrane potential because they're always in a state of depolarization or repolarization. Okay? Contractile cells, your heart only contracts if it's 60 beats a minute. All right, there's periods of time when it's relaxed. So that's when we see a resting membrane potential. Then it contracts. What stimulates a ventricular muscle cell or an atrial muscle cell to contract? Not pressure.
Voltage.
Voltage. From where? A current. Where's that current coming from? The autorhythmic cells or a previous contractile cell that might be upstream from it. Is that an action potential voltage or is that a graded potential voltage coming from the autorhythmic cells or from the ventricular muscle cells? Action. Good. It's an action potential. So we never see a graded potential on our autorhythmic cells. Because as soon as that current comes in from the autorhythmic cell or from the contractile cell's gap junction, it's created by previous action potentials. So it itself is strong enough to open voltage-gated channels. So this is our resting membrane potential, and this is a voltage-gated sodium channel opens, like our typical skeletal muscle cell. Okay, so the action potential opens because sodium voltage-gated channels open. And then they close. And normally we would have potassium voltage-gated channels open and bring us back down with repolarization. However, as the sodium channels are closing, we have slow calcium voltage-gated channels open. Are positive ions going to come into the cell or out of the cell? If it's calcium, they're going to come into the cell. Okay? So that's going to make the cell more positive rather than more negative. But we do have potassium voltage-gated channels. So we're back to that bathtub analogy. Now we've pulled the plug. The potassium ions are draining out of the cell. But we keep adding more water. So the bathtub isn't going to empty as quickly as it would if it was skeletal muscle cell. All right, so over here is the action potential of a skeletal muscle cell. That bathtub drains right away. But in cardiac muscle cell, because we're adding calcium while we're trying to lose potassium, it takes a long time and then finally the calcium channels close, and then we have this precipitous drop as we lose potassium. So calcium channels here are going to close. And then we repolarize. Why do you think we changed this for cardiac muscle? Why do we add those slow calcium channels to prolong the action potential? Look up there at the second diagram for skeletal muscle where it shows contraction related to the action potential. With the short action potential, the relative refractory period is over pretty quickly. When the absolute refractory period is over, we're into relative, and we can stimulate that skeletal muscle cell to contract again. Remember TREP and technique? So skeletal muscles can be stimulated to contract before they've actually relaxed. And they can be placed in a condition where they have a continuous contraction with no relaxation. What would happen to your blood pressure if we did that to heart muscle cells? And they just spasmed. Circulation would stop because we have to have a continual input and output of the heart. So having this prolonged plateau means that the absolute refractory period for cardiac muscle is almost as long as the contraction period. And that prevents another stimulus from occurring before the cardiac muscle is completely relaxed and prevents tachycardia. Yes? Do what? Okay, so we have a prolonged action potential. This entire event is the action potential. And that prolonged action potential of cardiac contractile cells prolongs the absolute refractory period. And that means, so here's our refractory period to here. And here's our contraction, so the heart started to relax again. And so that means that the heart cannot be stimulated to contract as long as we're in the absolute refractory period. So it prevents cardiac muscle from going into tach. You could suffer, a Charlie horse or something in your leg, but not in your heart. Okay. So those are the three membrane potentials that I want you to be able to relate to cause and what's happening at the membrane. Skeletal muscle, which is just kind of our reference, autorhythmic cells, and cardiac contractile cells. Okay, let's take a break till about 10 after. And when we come back, we'll look at ECGs, what's happening here, the conduction system. And then that'll take us about to a quarter of when we get back from break, about another 30 minutes. And then we'll do the heart dissections. Okay? I'm going to go run off the keys to the lab exam, and then after the second break, I'll give you back your lecture exams and lab exams.